This is a production of Cornell University.
Welcome everyone, friends, family, and colleagues. Jennifer Robin Wilson is originally from Charlotte, North Carolina. She received her Bachelor of Science in Plant and Environmental Sciences from Clemson University in Clemson, South Carolina. While at Clemson, Jenny led many student organizations, including founding the Clemson Agronomy Club, heading up the iGEM synthetic biology team. Jenny explored different research opportunities as an undergraduate, working in the labs of Drs. Huang Lao and Guido Schnabel, in addition to an REU internship with Dr. Sona Pandey at the Danforth Center. Jenny was recognized with numerous awards for her dedication to scholarship and research, most notably an NSF predoctoral fellowship. Jenny joined my lab in 2016 to pursue her PhD in plant pathology, studying molecular mechanisms of transmission of Poleroviruses. From our earliest conversations, I knew we would be a great scientific match. During her time at Cornell, Jenny has been active in the Plant Pathology Graduate Student Association, serving as its president and treasurer. Jenny has also been very active in outreach activities for children, especially young girls, through the Expanding Your Horizons program and Girl Scouts of America. She has also been the recipient of a National Institutes of Food and Agriculture predoctoral fellowship and the Barbara McClintock Graduate Student Award for unique and outstanding contributions to research. Jenny was awarded a SNPs Schmittu-Novak grant to pursue her collaborative research with Josh Chappie's lab. Over the past year, Jenny has been working closely with the cotton growers to translate her Polerovirus research from potato into cotton. She was awarded an unsolicited grant from Cotton Inc. to identify overwintering hosts of the cotton aphid, and the Gary A. Herzog Outstanding Talk at the 2020 Cotton Beltwide Conference. Jenny has been a true scientific partner during her time in my lab, bringing some of the most challenging projects to completion, as well as having her thesis chapters all published or just about ready for submission. Jenny's research led to the development of an invention disclosure describing a new method to control Polerovirus transmission by aphids. I am very proud of her and her achievements. She has been a beacon of knowledge for the newcomers in the virology program. Excitingly, Jenny was hired into a special USDA ARS postdoctoral fellowship program funded by ARS headquarters for projects themed high impact and a national grand challenge at the agency level. She will continue to pursue vector pathogen interactions research with Kai-Shu Ling, also a former Cornellian, at the US Vegetable Lab in Charleston, with the focus on Gemini viruses and whitefly interactions. But for now, please sit back and relax as Jenny takes you on a fantastic voyage through Polerovirus aphid interactions.
Thank you, Michelle. Thank you for that kind introduction. Thank you all for being here. I'm very excited to share my research with you all today. I hope you can see my screen. Before I get started, I want to point out there's on the right side of my slides, there's this red bar where you can put everyone's little faces on Zoom over there and it won't interfere with anything in the slides. So my dissertation research has focused on Poleroviruses. So you may be wondering, what is a Polerovirus? So these are viruses in the genus Polerovirus and the family Luteoviridae that infect plants, and they can cause many different destructive plant diseases. So shown here are 3 examples of Poleroviruses we study in our lab, and you'll notice that each one infects a different family of plant host and is transmitted by one or a few aphid vector species. So the main virus I focused on for my dissertation is Polerovirus, or PLRV for short. It infects host plants in the family Solanaceae, including its most important economic host, potato. And on some cultivars, it can cause this net necrosis of the tubers that you see here, thus making those tubers unmarketable. Its primary vector is Myzus persicae, the green peach aphid. Another important Polerovirus is cotton leaf rolled dwarf virus. This virus is currently emerging in cotton production in the United States. It infects cotton and other hosts in the Malvaceae family and maybe others, and its primary vector is Aphis cassipii, or the cotton aphid. Another important group of Poleroviruses are the cereal yellow dwarf viruses, which infect grasses in the Poaceae family, including oats, barley, wheat, and maize. And this particular one, RPV, is transmitted by Rhopalocyphum paddi, the bird cherry oat aphid. So my presentation today will focus on PLRV, which is short for potato leaf roll virus. So a little bit more about these viruses. So here is a schematic of what we think the virion looks like. So these virions have T=3 icosahedral symmetry. So that means that their basic shape is an icosahedron. And each face of this icosahedron is comprised of 3 protein subunits. Now, our particular viruses, they actually have 2 different proteins that make up their capsid. So shown up here is a diagram of the genome of PLRV. Its genome is RNA that's single-stranded of a positive sense. So that means it looks like messenger RNA and it can be directly translated into protein. It's only 5.8 kilobase pairs long, and it encodes 8 different proteins. So each of these boxes represents an open reading frame, which encodes for a separate protein. The virus structural proteins are encoded over here by these ones I have colored in blue and orange. So the majority of the virus particle, or virion, is comprised of coat protein, shown in blue. But at the end of the reading frame for coat protein, there's a leaky stop codon. So this means that usually the ribosome stops when it gets there, but sometimes it keeps going, and it adds this extension to the protein, which we call the read-through domain. And so this makes a longer protein, which sticks out from the capsid surface and is incorporated into the capsid in some unknown ratio, though most of it is still coat protein. In addition to virion assembly, these structural proteins have been found to have different roles in the virus's life cycle. For instance, the coprotein has been found to be important for aphid transmission and movement in the plant host. Through mutational analysis, it's been found that the N-terminal region of the read-through domain is important for aphid transmission. So virus particles lacking this domain are either poorly or not at all transmissible by aphids. And the C-terminal region of this protein has been found to be involved in phloem restriction. So very importantly, these viruses and their plant host are only found in the vascular system, specifically in the phloem. And this is believed to be a co-adaptation for transmission by sap-sucking insects like aphids. So to get successful transmission of these viruses, you need the interaction of 3 different biological players. You need your virus, vector, and host plant. And the interaction between virus and vector is particularly important for these viruses because aphids are their only natural means of inoculating a new plant host. And the way they're transmitted by aphids is quite interesting. We call it circulative transmission. So I'm going to give a brief overview of this transmission process. It all begins down here in the infected plant in the phloem tissue where aphids feed. And when they feed and they take up the phloem sap, they also ingest virions. And these virions must cross 2 important barriers in their insect vectors. And it passes each barrier by a method known as receptor-mediated endocytosis. So we're going to zoom in to what's happening at the first important barrier, the gut. So to orient ourselves on this diagram, at the top of this image, we're inside the gut in the lumen. And down here on the bottom, we're outside the gut or in the insect body cavity. And in between here is the cytoplasm of gut epithelial cells. So this process begins when the virions are ingested, and the virions are recognized by aphid receptor proteins up here on this top or apical membrane. Once recognized, they are endocytosed, and this is believed to be done via a clathrin-mediated process. And then they traffic through the cells of these guts using these vesicles, so membrane-bound vesicles. Sometimes the virions line up in a row and create this tubular vesicle you see here, and they traffic across the cell until encountering this bottom or basal membrane. They fuse with this membrane and are released into the body cavity of the insect. From there, they rapidly diffuse until encountering the second important tissue barrier, the accessory salivary gland, where it does everything all over again backwards, binding to that receptors on that bottom membrane, crossing those cells, fusing to that top membrane, and being released into the salivary duct of the insect, where the virus particles can be spit into the next plant host along with salivary secretions for feeding. So as you can see, this is a very involved process, and the relationship between Poleroviruses and their vectors is quite unique. Mostly because, as I mentioned, the virus depends on the aphid for its transmission. That's the only way this virus moves in nature. And it's actually been shown that these viruses manipulate their aphid vector to entice it to transmit them and to further increase its spread. The vector, on the other hand, it doesn't need to transmit virus, and there are energy costs associated with doing so, but there are also benefits to being a vector, and that's partly why this relationship is maintained. So one way that Poleroviruses manipulate their vectors has to do with vector attraction and dispersal. So when Poleroviruses infect a plant, such as potato, they actually cause that plant to produce volatile compounds that attract the aphids. It brings the vector in. And once the vector feeds on that plant, it acquires virus, and it has the virus inside its body. And interestingly, their preference actually switches after they've acquired virus, and now they prefer to go to an uninfected plant. So this encourages them to leave that infected plant and go find a new host to inoculate. And aphids are normally non-winged, but sometimes they produce winged forms after cues like crowding. So these winged forms of the aphids are sort of what's driving the movement of the virus in the field. Another thing that PLRV does for its vector is it actually increases vector fecundity and longevity. So the aphids have more offspring and they live longer. And living longer is very important for the virus because these aphids remain viruliferous for the rest of their lives. So once they have the virus, they will have it until they die. So if you make your vector live longer, that means that you're going to have more transmission over the lifetime of that insect. So that's one instance where the interest of these two players are aligned. The virus wants to increase the length of life of the insect, and it also wants to live longer. But sometimes there's conflict. And so sometimes these interactions actually involve a balancing act between competing interests of each biological player. So today I'm going to share with you 3 different stories of this balancing act. One is looking at an instance of vector manipulation. The other is vector defense. How does the vector respond to having the virus in its body? And the third has to do with virus structures. So close examinations of the capsid itself has given us more insight into these interactions. So the first story has to do with vector manipulation. So the manner in which these viruses are transmitted, circular transmission, is actually one of many different ways that insect vectors transmit their viruses. And these different modes of transmission exist on a continuum, from viruses that do not replicate and do not circulate and only transiently associate with their vectors, to those that circulate and replicate, which is a much more prolonged interaction. Aphids transmit a group of plant-infecting viruses by a non-circulative mode, the Poleroviruses, viruses in the family Polerovirus, and these viruses only bind the mouth parts of the insect and stay with them for only a few minutes, actually. On the other end of the spectrum, example viruses that replicate in the insect would be densoviruses, which are not plant-infecting viruses, they're insect-infecting viruses, but they do replicate in this insect host as well as circulate. Poleroviruses have been placed somewhere in the middle, so they definitely circulate in the insect vector, and they are believed to not propagate. However, recent work on other non-propagative but circulative viruses, such as the Gemini viruses, kind of cast doubt on whether they replicate or not. So we were curious to re-examine Poleroviruses. Are they doing some low level of replication, possibly? And this is really important because that really changes the nature of relationship. That changes a lot of fundamental aspects of the biology of this interaction, as well as potential management strategies. So we wanted to reevaluate this paradigm using a new technique called small RNA sequencing. And small RNA sequencing takes advantage of the fact that in plants and invertebrates, antiviral immunity is largely mediated by small RNAs. So here's how it works. When a virus is replicating in a host, at some point during its replication cycle, it will make viral RNA of a double-stranded nature. This double-stranded RNA is recognized by enzymes in the host called dicers, and they cut the double-stranded RNA into these smaller pieces, which are called virally derived small interfering RNAs, or viRNAs. And they're typically 21 to 24 nucleotides in length. A single strand of this viRNA is loaded into this protein complex called the RNA-inducing silence complex, or RISC, and they're specifically loaded into a protein called argonate. Argonate uses that piece of viRNA to find and destroy other pieces of RNA that are complementary to it. So this often results in the degradation of viral messenger RNA, and thus the downregulation of viral genes and slowing the replication of this virus in the host. So when we do small RNA sequencing, we take these small viRNAs, we sequence them, and we compare them to a database, and we say what viruses are present in our sample, and those viruses must all be actively replicating, and the host is launching an immune response to them. So in this way, small RNA-seq is a powerful tool to detect if a virus is replicating. So we not only wanted to examine if our aphids were launching an immune response to this Polerovirus, potato leaf roll virus, but we needed a negative control, which would be a virus that does not replicate. So we decided to use the Polerovirus potato virus. Why? Because it's also transmitted by our aphid, Myzus persicae, and it infects the same host, potato. So here's our experimental design. Aphids were exposed to a PLRV infected plant. They were also exposed to PLRV in a sucrose diet. So you can actually purify the virus from the plant and then spike it into the sugar solution, which the aphids will feed on. And then you can disentangle effects that are due to PLRV in the plant versus just PLRV on its own. We also expose them to PVY, so this is that non-circulative virus as a negative control, as well as our mock-inoculated healthy plant control. So aphids are exposed to these treatments for a few days, and then we move them to turnip plants for a very important step that we call gut clearing. So we're trying to see if we find small RNAs to PLRV in the aphid samples. But when they feed on a PLRV infected plant, that plant is also producing small RNAs to PLRV. So we had to make sure we cleared those out of the gut lumen of this insect so that when we sequenced it, we were only seeing aphid small RNAs. And turnip is a great plant to use for this because it's not a host of PLRV and PVY, so the virus can't replicate there and there won't be any small RNAs detected from those plants, or mapping to virus anyway. So we extracted the RNA from those insects and prepared them and sent them for Illumina sequencing. And when we got those reads back, we actually found that there were no reads mapping to PLRV in our aphid samples. So this shows us that the aphids are not launching a small RNA response to this virus, and this virus is likely not replicating in the insect vector. So this confirms that paradigm that these viruses actually do not replicate in their insect vector. But the story doesn't end there, because we also saw reads mapping to a different virus, Myzus persicae densovirus, which is a virus in the family Parviviridae. It has a single-stranded DNA genome and an icosahedral capsid. So this is an insect-infecting virus. And so we saw these reads and we thought, okay, are colonies actually infected with this virus? Because sometimes viral sequences can be integrated into genomes. And it turns out there are a lot of integrations from this virus in the aphid genome. So we want to make sure what we were looking at here. So to do that, we mapped these reads to the densovirus genome. So the top here is a diagram of that genome. This virus produces 3 non-structural proteins indicated with the letters NS, and it produces 2 structural proteins indicated by the letters SP. And down here is a graph that's to scale with this, mapping those reads across the genome. And these gray boxes indicate pieces of this DNA that are integrated into the aphid genome. And these dots represent the reads that we found in our samples that match this virus, and they're color-coded by treatment. So what you'll see is that these dots are all across the genome, that we're finding reads of all these different pieces of the densovirus genome, and not just in these regions where it's integrated into the Myzus genome. So that does indicates this is an actively replicating virus. Our colonies are infected. And the reason we didn't know that they're infected or it doesn't pose a problem is that this virus is actually very mild and it doesn't really harm the vector all that much. It only slightly reduces fecundity and lengthens their development time. But now that we have this actively replicating virus, we can develop primers to detect it. So we designed our primers right here in this intergenic region of the virus, so that when we use these primers, we're looking at genome copies of the densovirus and not just transcription from the integrations in the aphid genome. So our aphids are infected with the virus, and they're launching a small RNA immune response to that virus. Here is a snapshot of that response. So on the x-axis, we have the different sizes of small RNAs that the aphids are producing against this virus. And the y-axis is the relative abundance of each of these sizes. So first, let's look over here in this size range, 21 to 24 nucleotides. This is the typical size range of viRNAs, and it seems in our aphid samples that 22 nucleotides is the most abundant. However, we also saw an increase in these larger small RNA over here in this 33 to 40 nucleotide size range. And this size range does not correspond to any known class of small RNAs. We also noticed this was only happening in one of our treatments, which is when aphids are exposed to a PLRV infected plant, but we don't see this when they're exposed to the PLRV, the same virus given to them via a diet feeding system. So whatever is causing this is only happening in a PLRV infected plant and not just by the virus itself. So we don't know if this is incomplete dicing of transcripts, or maybe this is some alternate pathway that's being activated in these aphids that are exposed to the PLRV plant. But regardless, it seems that this immune response is altered. We're getting this different distribution of these small RNA. So we were curious if this altered response was as effective at controlling that other virus, MpDNV. So what we did is we exposed aphids to an uninfected and a PLRV infected plant and quantified that virus using the primers I mentioned earlier. And we found that aphids exposed to a PLRV infected plant did have significantly higher titer of the densovirus. So their immune response is not as able to control the replication of this virus. So that's interesting. What may be causing that? So when you think about viruses and small RNA and inhibition of the immune system, maybe your mind goes to viral silencing suppressor proteins. So these are proteins produced by viruses to inhibit some aspect of this pathway, to interfere with it somehow as a form of counter defense. So the protein encoded by Poleroviruses that does this is called P0. And the particular place that P0 intervenes is by marking the argonaute protein for degradation. So this is what happens in the plant host, but we were curious if the P0 protein could also be responsible for what we're seeing in the aphid vector. So to get at that, I needed to deliver this protein to the aphids. So I turned to a model host plant, Kochiana benthamiana, and I expressed this P0 protein in some leaves. I let the aphids feed on that protein, and then I quantified their amount of densovirus. I also had another protein as a negative control, as well as uninfiltrated leaves, and leaves expressing the entire virus as a positive control. And you can see that being exposed to just the P0 protein led to a significant increase in that densovirus titer, which indicates to me that P0 is at least partly responsible for this phenotype that we're observing. So we do not know whether P0 is getting inside the aphid tissues and it's interacting with things in there to cause this effect, or maybe this is an indirect effect of what P0 is doing in the plant. And work is ongoing to sort of disentangle and delve a little bit deeper into that mechanism. So while we are on the verge of finding a mechanism, what still remains is an evolutionary rationale. Why would PLRV want to do this? Why would it want to make its vector more susceptible to an insect infecting virus? So we looked a little bit more closely at the literature about densoviruses, and it's actually been found that they can cause aphids to produce winged morphs. So here is a paper from PNAS where they looked at a different aphid species, the rosy apple aphid. And so they had a normally non-winged lineage of this aphid, so a group of these aphids that did not produce wings. And after they were infected with a different densovirus, crowding caused them to produce these winged forms. So here's an instance of one of these viruses causing them to have wing morphs. This connection between wings and these densoviruses was further solidified by a recent current biology paper, where they looked at two different lineages of the P aphid. And they had some lineages that after a crowding queue, they would produce the winged offspring, but others after crowding, they would not. And so they actually narrowed down what gene is actually responsible for this difference in wing production. And it turns out it was a horizontally transferred gene from a densovirus that was just happened to be more highly expressed in these lineages that gave rise to winged offspring. So again, a different densovirus, a different aphid species, but the connection is still there. So we're curious if that connection was present with our own colony of aphids, with our species of densovirus and our species of aphid. So unfortunately, all the insects in our colonies were infected by this virus. So we weren't able to compare infected and uninfected individuals, but what we could do is collect winged and non-winged aphids and look at how much densovirus was present in each of those cohorts. And we found that the winged individuals had significantly higher densovirus titer than their non-winged counterparts, thus establishing that connection in our own system. So if exposure to a PLRV infected plant leads to a greater amount of this densovirus, and more densovirus means more wing production, then we would expect to find a higher proportion of winged individuals when aphids are feeding on a PLRV infected plant. So while we didn't test this own experiment in our hands, we saw in the literature that Fred Gildow had done a very similar experiment with a different Polerovirus. And so he found exactly that, what we've hypothesized. Aphids exposed to a Polerovirus infected plant had significantly higher wing morphs being produced after a crowding stimulus. In fact, Gildow found that the ability of the aphid to transmit virus was not related to the increased development of alitae or wing morphs. Thus, he concluded the response of the aphid must be to the infected plant itself, which is exactly what we've observed in our small RNA experiments. We only observe this change when they're exposed to a PLRV infected plant and not exposed to the virus by itself. So we've developed a hypothesis for what we think is going on in this system. After aphids are exposed to a PLRV infected plant, their small RNA immune system is altered. And this effect is at least partially mediated by the P0 protein by an as yet unknown mechanism. Regardless of how P0 is acting on the aphids, after their immune system is altered, there is a proliferation of this other virus. They're not as able to control MpDNV in their bodies, and they become more infected. This increase in MpDNV potentially leads to an increase in the production of winged forms of the aphid. And these winged forms can then go on and better disperse both viruses. So we believe this is an instance of vector manipulation. But again, why would PLRV want to do this? This is so risky to make your vector susceptible to viruses. Yes, some of those viruses are great and they cause increased wing production, but some of those viruses may be lethal. So in this particular instance, PLRV has to balance the additional benefits of having more winged vectors versus the potential detriments of the vector potentially succumbing to a lethal virus. So in this case, the benefits must outweigh the detriments to cause PLRV to have evolved to cause this manipulation in its vector. So that's my first story about vector manipulation. But the aphid is not passive in all this. It has a say in this interaction. And so my next chapter is about vector defense. So despite all that we know about this transmission process from electron micrographs, the exact proteins involved on the vector side of things still remains unknown. Only a few of them have been identified to date. So to identify more proteins in the vector that may be facilitating transmission, a very talented staff scientist in our lab, Stacy de Blasio, decided to use a technique called co-immunoprecipitation, which can capture protein-protein interaction complexes. So here's her workflow. She exposed aphids to a PLRV-infected and uninfected plant and gave them 48 hours to acquire the virus. After the 48 hours, she extracted protein from these aphids and was able to pull down the virus and interacting proteins using an antibody that recognized the virus conjugated to a magnetic bead. So this antibody pulls virus, and the virus pulls along all these aphid proteins with it that it's potentially interacting with. And then you can elute these proteins and identify them using mass spectrometry. So when Stacy did this, she found 8 proteins that were significantly enriched and pull downs from viriliferous aphids. So when you do these experiments, you have to compare how much of this protein did I find and pull downs from viriliferous aphids, shown in blue, as compared to non-viriliferous aphids, shown in black. And these proteins enriched in that viriliferous treatment are likely to be your interactors. So of the proteins she identified, she found a glycoprotein, a collagen protein, a heparin sulfate peptidoglycan, 3 different cuticle proteins, an immunity protein, C1QBP, and paramycin, a component of the cell cytoskeleton. And of these proteins, the one that was by and far the most enriched was C1QBP. So we decided to focus on this protein for further study. So what is C1QBP? C1QBP stands for complement component 1Q subcomplement binding protein, which means that it is a part of the innate immune response in humans, which is the complement system, which is a part of the innate immune response in humans. This protein also has other roles, such as in infection, inflammation, and apoptosis. It's predominantly localized to the mitochondria, but it's been found in other compartments of the cell, which is illustrated with some of these immunostaining images over here. So in these images, C1QBP is stained with an antibody and it's showing up green. There's a different antibody towards tubulin showing up red, and the nuclei are stained with DAPI, which is blue. So in these bone cancer cells up here, you see that C1QBP has this cytoplasmic localization, and this is consistent with localizing to the mitochondria. Down here in these skin cells, C1QBP is still green, but now it's on the cell periphery, and this localization is consistent with the plasma membrane localization. So it seems that this protein can do a lot of different things in a lot of different parts of the cell, but its primary role is in immunity. So I just told you how these aphids do not launch a small RNA immune response to PLRV. Then maybe C1QBP is part of a different immune response. So not small RNA, but maybe another response is activated. So yes, okay. So when you do these co-IP experiments, sometimes you pull down proteins directly interacting with your bait, which in our case is virus, but you may have a protein interacting to that protein. You may capture these tertiary and quaternary interactions. So we want to make sure these proteins were actually directly interacting, and we wanted to independently confirm this interaction. So we presented this data at a conference, and we were actually approached by Cecilia Tamborindegi, who's a previous postdoc in Stuart Gray's lab and now an associate professor at Texas A&M University. And she said, that's so interesting. You guys found C1QBP in your pull downs from PLRV viruliferous aphids. I found that protein in my yeast 2 hybrid screening experiments. So Cecilia did yeast 2 hybrid using proteins from the aphid Schizaphus gramenum, which is also an important vector of Poleroviruses. And so she mated the C1QBP protein from these aphids with yeast strains expressing the coproteins of 3 different viruses in the family Luteoviridae, including PLRV. And this blue color indicates a positive interaction between that protein with the PLRV coprotein, as well as these 2 other luteovirids that this aphid transmits. So this shows that C1QBP is actually able to directly bind PLRV, and it's interesting that there doesn't seem to be a specificity for viruses this aphid transmits. So aphid C1QBP can interact with coproteins of several different viruses in the family Luteoviridae. So now we confirmed that interaction in vitro. We wanted to see if there's an opportunity for this interaction to occur in vivo in our actual system. Are these proteins in the same place at the same time? So to get at that, I did immunostaining of whole mount dissected aphid guts. And here are some of my images. So I stained C1QBP protein using an antibody that recognizes this protein from humans, and it shows up as green in these images. So we're seeing it has a punctate cytoplasmic localization, and it seems to be inside this large network or matrix. The PLRV protein, on the other hand, I stained with a separate antibody, and this just appears as red in our images. So the PLRV also has a punctate localization, but it appears to be going to a different subcellular compartment. And so we don't have a robust set of cellular markers for hemipterans, for aphids. So we can't say for sure what compartment C1QBP is occupying, but we could say that the virus is potentially in those membrane-bound vesicles that were seen in those EM studies. And the places where these two proteins come together will show up as yellow. And so we have a few places where it seems these two separate compartments contact one another, such as here and here. And here's a zoomed in view of that here, these two compartments coming together. So that may allow the opportunity for interaction. But a second category of co-localization we observed was along the cell periphery. So this is, presumably an aphid gut epithelial cell, and the signal we're seeing on the outside here is that strong yellow co-localization signal between these two antibodies. So here's a zoomed in where you see these two proteins seem to come together along the cell periphery, which could potentially be the plasma membrane. So we've sort of had this multifaceted view of this interaction. We found via two different methods that this protein interacts with PLRV. One of those methods indicates a direct interaction, so the yeast-2 hybrid. We've shown that they potentially could come together in space and time in the aphid guts to actually have that interaction occur. But obviously there are other tissues in the aphids that are involved. So there could be interactions occurring in the blood of the insect or maybe the accessory salivary glands. Regardless, it seems that these proteins potentially do interact in the aphid, but what is it doing? What is the function of C1QBP and why is it binding virus? So to get at that, I actually was lucky to find that there was a chemical inhibitor of this protein already published, and we were able to get that molecule from the authors of this paper shown here. And so I wanted to use this inhibitor to inhibit the C1QB protein in aphids and then see how well they transmit virus. So to do that, I again turned to a sucrose diet system. So I spiked the inhibitor into this diet and I allowed aphids 48 hours to feed on it. And then I took them through a transmission assay. So I gave them a source of virus. So these are PLRV infected leaves. I gave them 24 hours to acquire virus. And then I gave them healthy plants, which they could inoculate for 72 hours. And these are potato seedlings. After 3 days on the seedlings, the aphids were removed by a pesticide application. And then I kept each of these plants to see if it would become infected with virus. And that gives me a transmission efficiency. How many plants became infected out of those I inoculated? So here are the data. On the x-axis here, we have the different concentrations of this inhibitor that I tried. So 50, 100, and 200 micromolar, as well as a 0 micromolar negative control. And then on the y-axis, we have the transmission efficiency. And I actually found no significant differences in the rate at which aphids transmit virus after each of these different concentrations of inhibitor. However, I looked more closely at the plants that did become infected in each of these treatments, and I found an interesting trend. Titer in the recipient plant increases with increasing inhibitor concentration. So again, on the x-axis, we have my different inhibitor concentrations, and now on the y-axis, we're looking at titer, how much virus is present in these plants. So for the 0 and 500 micromolar treatments, we have lower titer than the 100 micromolar of the inhibitor. And at the 200 micromolar dose, we see that this difference in titer is now significant. So what does this mean for the role of C1QBP in transmission? So as you recall, this is an inhibitor, so inhibits the function of C1QBP. And it seems when we do increasing amounts of inhibitor, we are also getting increasing amounts of PLRV titer in these recipient plants. So this to me suggests that maybe C1QBP is somehow a negative regulator of PLRV transmission. So its normal function is to either lower the amount of virus these aphids are inoculating into these plants or that's accumulating in the insect body, possibly. But why would C1QBP do that? What could be, it's a negative regulator, but what does that mean for the function? So I hypothesize that maybe the role of C1QBP in immunity may be conserved. So complement receptors in humans, such as C1QBP, have been previously shown to recognize pathogen-associated molecular patterns, or PAMPs. And this C1QBP protein itself has been found to bind several viruses in humans, such as rubella, HIV, and herpes simplex virus. So we've shown that, C1QBP from Myzus binds PLRV, C1QBP from that other aphid binds 3 different Poleroviruses. So it seems that maybe this protein is recognizing viruses more generally. So is it possible that C1QBP could be acting as some sort of pattern recognition receptor in aphids? But again, why would they, if they're doing that and they're playing this immunity role, why would aphids launch an immune response to a virus that doesn't replicate? So I wanted to go back to this electron micrograph from some of the early studies of transmission, where sometimes they see virions in this lysosome-like structure in the aphid gut cell. So here are the virions here. So it's possible that maybe the aphid wants to regulate how much virus is in its body, and it may be degrading some of these virions. I don't know if C1QVP is involved in that process. It's just an hypothesis. But it's possible the aphid may want to regulate how much virus it transmits, so that the benefits it gets from being a vector, such as increased fecundity, longevity, will outweigh the energetic cost of actually moving those virions through its body. So I think this is, again, one of those examples of a balancing act each of these biological players must make in this interaction. So that was my story about vector defense. This third story, we're looking at virus structure. So prior to my dissertation, there were no known structures of Polerovirus capsids. And why that is may have to do with protein disorder. So what we're looking at right now is a disorder plot. So on the x-axis, we have the different amino acid residues of this protein. And at the top, I have indicated the different domains of this protein. And so this red line here indicates about half disorder, 50%. So anything above this, we can say it's mostly disordered. So you can think of that as floppy. And anything below this line has a more ordered regular structure and are more probable candidates for structure determination. And it actually turns out these different regions of disorder may have important roles to play in virion assembly. So this piece right here is what we call the R domain, and it associates with the viral RNA that's the capsid wraps around. So this disorder may be necessary for it to form those interactions and assemble the virion. This peak right here, all this disorder we're seeing in this region right here, we call this the proline hinge, because this is where that capsid protein connects to that protruding read-through domain. So it may need flexibility in that region to form this three-dimensional structure. And this region of the protein, the C terminus of the RTD, is highly disordered, but it's actually believed to be at least partially cleaved off in the assembly of complete virion. So this may not actually be present in the completed mature capsid. So these two regions are ordered though, so maybe they could be used for structure determination. So actually a recent cryo-electron microscopy structure was published for the coat protein down here, and we actually decided to focus on the N-terminal portion of the read-through domain down here. And we worked with a group of structural biologists here at Cornell, specifically with Josh Chappie in the Department of Molecular Medicine, and they decided to go after the structure using X-ray crystallography. So I worked closely with Carl Schiltz, a former graduate student in that lab, and he was actually able to successfully express and purify this protein from PLRV. So shown here is an SDS page gel, and we see large quantities of that protein there. He was able to grow crystals of this protein, and he used X-ray diffraction to solve the structure of this protein. So here is the structure. This protein forms actually two different structural domains. So down here in the bottom in pink, I have the P domain. We called it the P domain because it protrudes from the capsid surface, and it's also homologous to the P domains of other plant infecting viruses, which also protrude from their capsid surfaces. The fold of this domain is what's called a jelly roll. So that's two beta sheets wrapped around one another. The second structural domain we saw is up here shown in blue. We call it the cap domain because it sits atop the other domain. And the fold of this domain is a beta barrel. So that's several beta strands twisted around one another. So it's not unusual to find two different structural domains in the same protein, but what is unique is how they are oriented to one another. So now what we're looking at is a topology diagram. And this is if you were to take the protein and map it from start to finish, N terminus to C terminus, and draw in every secondary characteristic, structural characteristic you came across. So these arrows represent the beta strands that we see over here. And this diagram is color-coded the same way. The P domain is pink and the cap domain is blue. And so interestingly, we see that the pieces of the protein sequence that encode the cap domain are interspersed between the pieces of the sequence that encode the P domain, which is quite unusual because usually when you have two domains, they're linear in the sequence, one right after the other. So this protein has quite a unique topology. Another interesting feature of this protein is actually that it forms a dimer. So we're looking at a monomer here, but when you put the two monomers together, you get a dimer like this. So we still have the P domain down here in these shades of pink and the cap domain up here in these shades of red. And the dimer is glued together by the piece of this protein that we call the C peptide because it's from the C terminus of the protein. And so this protein only forms a dimer in solution. It also formed a dimer in the crystal, and we believe it forms a dimer on the virion surface as well. So we can model how we think it sits atop the capsid. So down here are those coat proteins, and here are the two N-RTD monomers coming together over the capsid. And because they have to be a dimer, we predict that they are present on the twofold rotational axis of symmetry of the virion. So let's look a little bit more closely at this dimer. So this is the same dimer, but now it's in a space-filled model. And we've color-coded it based on the conservation of each of these residues. So the residues colored in pink, every virus in the family Luteoviridae has the exact same residue at that position. Residues shown in white are a little more variable, and those shown in blue are the most variable. So different members of the family may have different residues present at that position. So let's focus on the cap domain. So we take the dimer and we look at it top down. This is what we see here. So I'd like to draw your attention to these pockets that are in this dark, dark shade of pink. And these are residues absolutely conserved across members in this family. Every virus has the same residue at this position. And so that would point to some very important role for these residues. And so because this protein has been found to be important, this piece of the virus capsid has been found to be important for aphid transmission, we hypothesize that maybe this is part of the site where aphid proteins bind here, maybe aphid receptors. Because previous mutational analysis has found that if you delete this portion of the protein, you can't get transmission. So another reason why we think this cap domain in particular is important for transmission is where we find it in other viruses and where we don't. So the closest structural homologue to the N-RTD is viruses in the family Tumbospheridae. And so I'm showing here superposition of the P domain of one of those viruses, melanocrotic spot, with the PLRV P domain. And you see that they line up almost perfectly. However, this Tumbos virus, MNSV, has no equivalent to the cap domain. The cap domain is unique to PLRV and Poleroviruses. And so we may ask, are these other viruses that have this P domain, are they transmissible by aphids? And the answer is that no, they are not transmissible by aphids and they do not have the cap domain. So that to us is another piece of evidence of the role of the cap domain that may be important for aphid transmission. So this structure has given us a lot of hypotheses, but how can we test them? So what I'm showing here is a simplified schematic of how the virion may facilitate binding with aphid receptors. So coprotein is again shown in blue, the N-RTD is these orange spikes, and the aphid receptors are these purple Pac-Man down here. And I say simplified view because the coprotein has also been found to be important for facilitating with these receptors. But, you know, if the N-RTD does play a large role in binding to aphid tissues, what if we just flooded those tissues with the purified form of this protein? What if they plugged up those receptors so that when the virion came along later, it was not able to bind and we get a knockdown in transmission? And if that works, then maybe we can make mutations to this protein, the purified form of this protein, and maybe it would no longer be able to bind these receptors and knock down virus transmission. So let's test the hypothesis. So I needed to deliver this protein to different aphid tissues of interest. So one of those tissues was the accessory salivary gland. And so I delivered the protein to them using a technique called microinjection. So when you do this, you take a microcapillary needle and you stab the body wall of the insect and you inject the protein, bypassing the gut and giving it access to the accessory salivary gland. And then to deliver the protein to the gut, I used our tried and true method of sucrose diet delivery. So again, that they ingest the protein along with their diet and it goes straight to that gut tissue. After I exposed them to this protein, I wanted to test their ability to transmit virus. So I gave them a source of virus and I gave them healthy plants to inoculate. I kept those plants and calculated a transmission efficiency. So let's look at the microinjection data. So I had a no protein control. So this was just the buffer that I injected the aphids. I also had a different protein I wanted to test, BSA, bovine serum albumin, which is completely unrelated to transmission. So with this treatment, we're just asking what happens when you inject aphids with a protein. And it turns out it does not change their transmission to inject them with BSA, and nor does it when you inject them with the N-RTD. So I saw no significant differences in transmission from this experiment using this delivery method. What about delivery via the diet? So again, I had a no protein control. So this was just the plain sucrose diet. I had my BSA protein control, so this other unrelated protein. Interestingly, I saw an increase in transmission when aphids feed on the BSA protein, which is actually something already reported in the literature. They found that this happens when you use diet feeding for BSA and any number of other proteins that have nothing to do with transmission, which is kind of, an interesting finding, but it was great to see that I could recapitulate a phenotype observed in the literature. So what about our protein of interest, the N-RTD protein? When aphids are exposed to the N-RTD protein, it actually significantly decreases their ability to transmit PLRV. So this confirms our hypothesis that the purified form of this protein can inhibit transmission of the virus. Great, so we have our proof of concept. Now we can move forward and make mutations and test hypotheses about the structure. So we made two different categories of mutations. We either made single alanine mutations, or we had this one form of the protein that we mutated 3 residues at once because the residues were closely together and they formed a cluster. So for the single alanine mutations, we mutated this one, this one, this one, and this one, 1, 2, 3, and 4. And remember, this is a dimer, so they're also present on the other side. And the reason we chose these residues is because they're on the cap domain, which we think is important for interaction with aphid tissues. They were all surface exposed, so that means they're in a position where they could potentially interact with aphid proteins. And all of them actually had charged side groups, so maybe they could be involved in electrostatic interactions with the vector. So we made these alanine mutations, which makes them uncharged, and I put them through my bioassay. I let the aphids feed on them for 2 days. I gave them a source of virus. But then when I went to move them to uninfected potato seedlings, I saw that all the aphids were dead. So I repeated my experiment, and they were dead again and again and again. So I decided to quantify the death, which is what we're seeing here. So on the x-axis, we have the different treatments I exposed them to, and the y-axis is how many aphids died. So again, we have a no protein control and BSA, both of which caused very little, if any, mortality. We had the wild type form of this protein, which caused a little bit of mortality. And then we had these 5 different mutant forms. So mutants 1, 2, 3, and 4, as well as the cluster. And so we found that all except one mutant form actually caused significant mortality in these insects, with mutants 4 and cluster causing near complete mortality. So this is quite unusual. Why is this happening? Why is changing one residue of this protein causing these aphids to die in such high numbers? So if you think about how this protein binds aphid receptors, or maybe it binds other aphid proteins, what if it were to bind them too tightly and those proteins couldn't perform their normal functions? And that could potentially lead to the death of the vector. So that's one hypothesis we have for what may be occurring. The virus has this balancing act between, I need to bind to these proteins to get through the aphid, but if I bind them too strongly, they can't perform their normal functions and I may kill my vector. So this to us may indicate that the wild type form of this protein is fine-tuned to balance these perfectly. It binds with the right affinity to be transmitted, but not kill the vector. So that is my third story about the balancing act between these two biological players. So I've shown you 3 stories, one about vector manipulation, where the virus knocks down the antiviral immune system of the vector. I showed you one where the vector may fight back and regulate the amount of virus in its body. And the third story was about how the virus structure itself may be fine-tuned to not kill its vector. So this is all exciting, but how can this basic research lead to solutions for agriculture? So I want to revisit that finding. The feeding aphids on the N-RTD protein can knock down transmission of PLRV. It turns out this is a highly reproducible phenotype. I did it many times, and I performed a meta-analysis, which is a statistical analysis used to test the efficacy of drugs or pesticides. And I found that when you pre-expose the aphids to the N-RTD, it reduces the chance of the plant become infected on average by 44%, with efficacy ranging from 22 to 88% reduction. So it seems that this may be a method that we could use to potentially control the spread of this virus in the field. But what about other Poleroviruses? Is this something unique to PLRV, or could we apply this to other systems? So the Chappie lab has been very busy solving N-RTD structures for us. And so they've actually already solved 2 other Polerovirus N-RTD proteins, one of which is shown here. This is the N-RTD protein of turnip yellows virus. And it should look very familiar. You'll see it has the same P domain and the same cap domain. The one thing that may look a little different here is this C peptide, but actually they were able to get a really high-res structure for this virus, 1.5 angstroms, so they're actually able to solve more of the C peptide. So this is just more of what we would have seen in PLRV with higher resolution. And if you were to superimpose these 2 N-RTDs, you see that they line up almost perfectly. So it seems that the structure of this protein is highly conserved across Poleroviruses, which is great news because it means what we found in PLRV may also be applied to other Polerovirus systems. So I wanted to translate this finding we found in the lab with this diet feeding system to maybe something we could deploy in the field. What if we could get the plant to make the protein for us? So to test that, I again turned to the model plant, Nicotiana bismiana, and I transiently expressed these proteins. So I expressed the N-RTD protein as well as a negative control protein, and I had my uninfiltrated control. And then I took these aphids through the transmission assay and quantified their ability to transmit PLRV. And again, I found that the N-RTD protein preliminarily does slow their transmission of PLRV. So it seems that this delivery strategy may work. We could get the plants to express the protein for us. So work is ongoing to move this protein into our important crop host, so to transgenically express it in potato. So we've worked with Joyce Van Eck at BTI to transform potato for just that. And we're rapidly translating these findings to the new emerging Polerovirus cotton leaf-rolled dwarf virus. Can we use the N-RTD as a new novel control strategy for that emerging pathogen? And we're not just looking at using the wild type form of this protein, but the mutant forms as well, which may actually serve as a novel pesticide. So we're hoping to add some new tools to the toolkit to manage this economically important group of viruses. So with that, I would like to acknowledge a lot of people because it really takes a village to make a PhD happen. I'd like to thank HEC Lab members, past and present, my wonderful collaborators, support staff, my special committee, and funding sources. Those shown in red are my co-first authors who contributed greatly to this work. But my time here hasn't just been about science, so I'd like to also thank my friends and family for all the fun times and their emotional support. And also, you know, a PhD is kind of a culmination of, you know, a lifetime of learning. So I actually want to take a moment to thank all my previous mentors, none of who are on the Zoom call today, but just the people who instilled in little Jenny a love of learning and a love of knowledge and helped me become the scientist I am today. And with that, I will take any questions via chat. Thank you. All right, chats are rolling in. Oh my, okay, Adam starts with a doozy. So Adam Bogdanov says, great work, Jenny. Oh, I didn't have to read that part. Because the N-RT dimerizes could reduce transmission in the feeding/competition experiment result from destruction of the capsid-bound N-RTD. Can you get heterodimers with the N-RTDs of the Poleroviruses it prevents transmission of? Any potential for engineering the plant receptor to ignore N-RTD? Great question. So yeah, I think there is definitely could be some interaction between the dimer, you know, the purified form of this protein and the actual virion form of this protein. So there's actually some interesting work out of one of our collaborators in France where they showed that there's actually a piece of the N-RTD that associates in trans with the virion. So you have the pieces that are associated with the virion, but also the soluble form may actually form associations with the virion as well. So that's totally possible as a explanation of what's happening. Sorry, let me close my Slack channel real quick. Okay, and as to your other questions, so we haven't tried doing any heterodimers with the other N-RTDs. That's a very interesting question because, yeah, I mean, how conserved is it, right? How conserved is the dimerization? And any potential for engineering plant receptors. So we have lots of great ideas for how we want to engineer things in the system. And so plant receptor, I mean, there are lots of plant proteins these viruses interact with. So there's a lot of potential there. As far as the aphid receptor, we still don't know the aphid receptor for Myzus persicae and for PLRV. So it's going to be really exciting once that receptor is identified and maybe that structure comes out, to see how it correlates with our structure and maybe can further explain some of the phenotypes we're observing. Okay. Okay, we have a question from Mariko. I have a question about your N-RTD mutants. Did you try feeding them to any other species of aphid, especially species that don't transmit PLRV? I did. So I fed some of these N-RTD mutants to the cotton aphid, and which does not transmit PLRV. And interestingly, it killed them too. But it wasn't, the mortality rates were not the same. So what was interesting to me is like one of the most lethal mutants to Myzus was not that lethal to cotton aphid. So, you know, that's an interesting finding. And it makes me wonder to what extent, you know, the N-RTDs seem to be extremely conserved, but how conserved is that aphid receptor that, you know, the N-RTDs of a completely different virus may be able to bind them. So that's also makes us think about the biocontrol potential for these mutants as well. I see lots of congratulations. Oh, a new question. Yes, so we have a question from Igor. He says, based on the charge change of those N-RTD mutants, is there a way to test in vivo if they bind tighter to the aphid protein? I don't know about in vivo. There are definitely in vitro binding assays you can do, but a lot of those require you to know what your binding partner is. So that's part of one of our limitations here is we don't know the aphid proteins it's binding to. We do have plans to do like co-IPs with the N-RT protein, say what is it binding to in there. And we also want to do some microscopy to see, you know, where is N-RTD going in the gut? Are we seeing more binding, you know, qualitatively with these mutants versus the wild type forms? So excellent, excellent question. Okay, well, I don't see any more. So wait, oh. Okay, I got one. So Chris asks, are there known receptors, host receptors in the gut that mediate virus endocytosis? Yeah, so there are some known for other systems. So for a different virus in the lutiviriid family and the P. aphid, they found a protein called aminopeptidase N as a putative receptor. For Myzus persicae, they found a protein they think may be a receptor for some other Poleroviruses, so for that turnip yellows virus, and then another one, cucurba aphid-borne yellows virus. They think they actually have the same receptor, which is quite interesting, and that protein was called epherin receptor. But one of our huge limitations in this system is our functional assay. So people keep finding these potential receptors, but we're really struggling to actually validate them. So that's why all of these receptors are only putative at this point. Yeah, so Claire has a great question. So she said, you showed that the virion interacts with an aphid protein and that can influence transmission. Could the impact of PLRV on aphid behavior after acquisition be mediated through similar mechanisms? You confirmed that this virus does not replicate in insects, so she's wondering how changes in aphid behavior occur. Yeah, it's an interesting question. So I think our work with the P0 protein shows that virus proteins produced in the plant may somehow have activity in the aphid, whether they're getting in or not. So that's one way, PLRV can cause these changes in the aphid without replicating in them, is like using the plant host to make the proteins that it uses for that manipulation. Very interestingly, that finding that the aphid preference switches to uninfected plants after acquisition, they tested that using purified virus as well, and it still happened, which is very amazing to me. So seems like somehow in that case just the virion is involved, and I don't entirely understand how that could happen, but maybe the structural proteins are somehow involved in that. Okay, well, I think we're a few minutes over time anyway, but thank you all for coming. I see hand clapping. I guess, are they allowed to unmute themselves to applaud? I feel like I'm talking to a computer here.
Yay! Good job, Jenny.
Good job.
Congratulations.
Thank you. All right. Well, I have a defense to get to.
This has been a production of Cornell University, on the web at cornell.edu.